All right. Hello, everyone. My name is Shay Sullivan, and this is my humble attempt at drawing a eukaryotic cell. So I'd like to start with around the yellow line, the edge here. This is the plasma membrane, and it provides an important role in shaping and protecting a cell, um, helping to facilitate communication between cells and then neighboring cells, and also managing the semi permeable nature of the plasma membrane, so allowing certain particles in and out um, through passive or active diffusion. So this can sort of the plasma membrane is really important in helping to maintain homeostasis of a cell. And then, so in the middle here, we have the nucleus, sort of the lighter blue line. And the nucleus is often visible with a light microscope, and it's encapsulated in the nuclear envelope that's visible sort of at the bottom, trying to show a three-dimensional shape to the nucleus since they are typically round or spherical. And some cells, depending on their size, might have multiple nuclei. And the function of that is DNA storage, the function of the nucleus. But within the nucleus, there is a nucleolus, which is more purple there. And the nucleolus manufactures ribosomes. So the nucleus can really be thought of as really the blueprint to our body. So this cell might be more active and have more energy requirements. So there's multiple mitochondria here, and they can actually be different shapes. So we have sort of the oval or oblong mitochondria, and then maybe sometimes they're more kidney shaped, and the striped structure that makes up a mitochondria is actually called cristae. And mitochondria are highly mobile, and so that might be a reason for their shape change. So mitochondria take different starting materials and synthesize them into ATP, and because they help to synthesize all that energy, they're often considered the powerhouse of a cell. So some of the different starting materials that I read about include like lipids, starches, or sugars. So it can just really take just a different variety and turn it into something useful for the cell. So next, I would like to move on to lysosomes, which are labeled with little L's there and there, and they're spherical and enzyme shape or enzyme filled. And lysosomes are produced by the Golgi complex. And so they like to break down proteins and can sometimes be considered the recycling center of a cell. And then we have peroxisomes, which are labeled with P's because they look just like lysosomes oftentimes. And they're produced in the endoplasmic reticulum. And they often break down fatty acids that the mitochondria can use for energy, or they'll use molecular oxygen to oxidize organic materials, which produces hydrogen peroxide. So I read that they are sort of important for homeostasis then, if they're sort of helping to break down materials that can't be used by a cell and that kind of stuff. So moving on, we have the endoplasmic reticulum, which is demonstrated here. So they're interconnected channels called cisternae, and there's two kinds. So we have the rough endoplasmic reticulum, which is our dark green, and that's spotted with ribosomes, which are the little black spots. And the rough ER is continuous with the outer layer of the nuclear envelope, like we pointed out before. And then so the rough ER produces phospholipids and proteins for the plasma membrane, and it can be identifiable because they're sort of fingerprint shaped, like we discussed in class and really run like parallel to other channels of rough ER. And then we have our smooth endoplasmic reticulum, which is demonstrated with our light green color. And it's continuous with the rough ER, but it's more tubular and it doesn't have ribosomes. And smooth ER is abundant in cells that engage in detoxification, so cells in the kidneys or liver, since it can quickly detoxify like alcohol and other drugs. All right, so moving on, we'll go over to our orange Golgi complex, which often is compared to a stack of plates or a slice, like stacked slices of pita bread. And so the Golgi complex receives newly synthesized proteins from the rough ER, and then it can add finishing touches. And so it'll sort of like sort, cut, and splice some proteins or add like carbohydrate properties to some products. And it'll package proteins for transportation through the Golgi vesicles. So those are our little dots there. And so the Golgi vesicles, they just bud off of the rim of the cisternae, and they may become lysosomes, or they may fuse with the plasma membrane and contribute fresh proteins and phospholipids to that membrane, or they may become secretory vesicles that store a product for later release. Okay. So now we've talked about ribosomes a little bit and how we can see ribosomes on the rough ER and how they're manufactured in the nucleolus. But you can also have ribosomes, sort of free ribosomes that just are floating around the cytosol, and they're very little. And their function is to read coded genetic materials and assemble peptides and proteins. So they're clearly very important for such a little size. And then last but not least, we have a humble attempt at centrioles. So they are short cylindrical assembly. They're a short cylindrical assembly of microtubules arranged in 9 groups of 3 microtubules each. And so it's a paired organelle, and they lie perpendicular to each other. And then, so the centrioles play an important role in cell division. So they ensure structures in cytosol are arranged correctly for proper cell division. So to me, I guess that sounds like they make sure that cells are equal so that they can divide properly. They couldn't divide properly if they might have mutations, or they would be unhealthy cells if they don't divide properly. So we also have some pumps or channels in our phospholipid bilayer, and we have a very basic cell depicted here. Thanks.